Okay, fantastic. Well, welcome everyone to our panel this afternoon at the end of day one of the PDDP East Coast Summit. Our panel the title is Accelerating Your Drug Discovery Project with AI and Breakthrough Technology. And we have an incredible group of panelists today who are experts in AI and other breakthrough technologies. They include chemists and computational scientists and biologists who can all provide a broad perspective of how AI can accelerate drug discovery. So let me just introduce myself. I'm Tina Rogers. I'm not in AI or drug discovery. I'm actually in preclinical drug development. I'm a preclinical toxicologist. So this is not my area of expertise, but I've learned a lot already, and I'm excited about the panel today. We have some really exceptional, enthusiastic and passionate scientists on the panel today. So I just want to give each one of the panel members a chance to introduce themselves and just say a sentence or two about their the company that they work in and the role or how that relates to AI and drug discovery. So let me start with Elmar.
Hi, my name is Elmar Normandov. I am faculty at John Wayne Cancer Institute. My research lab is focused on experimental therapeutics, particularly on Wnt beta-catenin signaling pathway as it relates to cancer and cancer cell signaling. So to parallel with that, we're also developing methods that benefit early drug discovery. As I talked this morning, one of those methods is cellular target engagement. And basically, we have two basic lines of research. So hopefully in the future, that will expand further.
Great. Andrea?
Hi, I'm Dr. Andrea Small-Howard, and I'm the Chief Science Officer, and I'm on the Board of Directors at GB Sciences. GB Sciences is a phytomedicine company where we're developing multi-component drugs based on plant-inspired materials in a way that is leveraging molecular synergies among the components that are originally derived from these plants. And we're talking today a little bit about the fact that we've been able to accelerate this development using AI-enabled and machine learning technologies.
Great. Ashwini?
Hey, thank you, Tina. It's a pleasure to be on this panel. I'm the business lead for AI-enabled drug discovery at Millipore Sigma. Millipore Sigma Mercaga is quite established company in reagents and all of that. So needless to introduce the company on that front. But the company is taking strong foot forward in digital drug discovery solutions, including one of the solutions like Cynthia that I worked on before, the retrosynthesis design. So many such software solutions that are enabling R&D scientists to take their make their drug discovery much faster. And so I'm in the strategy and execution for cheminformatics solutions from Millipore Sigma, and I've worked on Cynthia before this. I'm a trained organic chemist and a photobiologist, so very much thank you for the introduction and saying we're all passionate group of scientists who always want to make a difference. So definitely thanks, and happy to be part of this group.
Great, thank you. Manish?
I always do this. Too many meetings where I keep myself on mute. But yeah, thanks again, Tina. It's good to see you again. So I'm Manish Pinglay. I head the business development team for discovery services at Sci Life Sciences. We're a CRO that's a full-service CRO that provides discovery services all the way from exploratory biology through to getting to a clinical candidate for our customers and our clients. We're deploying AI tools as well. So we have a computational group that deploys AI tools to help design molecules, to help synthesize molecules. So I'll speak a little bit about that. Also trained medicinal chemist by background, some biology expertise as well, but then helping other people do their science these days rather than doing it myself. So good to be here.
Great. Ranga?
Hi, thanks, Tina, and thanks to the organizers for this for the opportunity to participate in this panel. I'm Ranga Sundararajan. I'm the Chief Scientific Officer and SVP of Clinical and Translational Sciences at Berg. Berg is a clinical stage biopharmaceutical company, but what we core expertise is literally mapping the human biology at a molecular level and using AI and ML to gain insights between the differentials between normal and disease, and use that information across the entire clinical development spectrum, fundamentally tagging a parallel development of drug discovery and development towards biology-based development in the clinics.
Great. And Stephanie?
Hi, everyone. My name is Stephanie Scores. I work over at Chimera Therapeutics. We are a company that focuses on making protein degraders. I am a trained biologist. However, Chimera is a heavy biochemistry-focused company. So I am very familiar with AI technologies and how they can help characterize lead molecules. So very excited to be a part of this panel.
Great. I'm going to attempt to share just a brief slide just to show an outline today. Well, can you guys see that?
No.
Oh, yeah, I'm not sure why this is not working.
If you want to hit the...
Sam, can you help me out a second?
Yeah, if you want to hit the screen share button at the bottom beside the webcam icon.
Sorry, I couldn't hear that.
It's right beside the webcam icon, the camera icon rather. It's a screen share. It looks just like a screen.
Yeah.
And you want to
Yeah. I see that.
I'm going to ask you for two. Either I'll ask you to share your entire window or a specific window. So
if I tell it, it doesn't give me the choice that I want. If I do entire screen.
Yeah, do entire screen. Click on screen one and then click share. And it should work.
Yeah, it's not. That's okay.
So I think I ran into the same problem in the morning. So if you have your materials on two different screens, you want to put them on one screen, this screen and your shareable material on the same screen, and then share, and then basically you will have the PPT live.
Yeah, this says permission to use screen share is missing. Okay, anyway, it's not that critical. So I just wanted to basically go over the outline for today. So we're going to talk about, first we're going to talk about data, because data are the foundation of AI and machine learning. And then we're going to talk about how two basic categories of use of AI and ML. The first is to better understand your disease. And then the second is to design and optimize novel drug candidates. And then finally, we're going to talk about future trends related to AI and ML and other breakthrough technologies. Okay, so pretty simple, and it's very flexible, and this is going to be a great discussion. So there's going to be no rigid framework put around it. So let's start with data. So Rohit, what happened to Rohit? He was going to be our data person. He was here earlier, wasn't he? So Ashwini, maybe I'll ask you to talk about data first, and the importance of clean data and how you go about and data engineering and et cetera.
Sure. Yeah, I did see Rohit. Maybe he might have had some issues or so. Data. I think data is, as everybody would say, data is gold, data is key, data is all of it, right? And that's where it starts. But I think having clean research data is really radically important to reduce this uncertainty between early and late stage drug discovery and development. And there are two ways I've seen people address this problem. One, the startups have really taken this by the horn, is generating this data that is specific to their models by creating experiments that address that specific question. And so creating those experiments, creating that clean data set, whether it is rare disease, it's therapeutic area driven, it's question driven, but creating that and enabling the machine learning models to be trained. This has been a constant, I think, problem with large pharma companies that sit on a large amount of data, but it's not really usable because either the experimentation is not constant, so on and so forth. And so you can see the startups really catching up on this and being able to design drugs much faster than a large therapeutic companies because of their closed loop integration of experimentation with the data that they're generating, and then using that data to train the ML model, and keeping that a really tight net integration, a closed loop integration is key. So that is my overall, I think, take within the industry on data, how critical it is, how startups are tackling it. But there's another concept that I'm a fan of, and I would like to see this, I think, has been utilized in some of these consortia. Is called the federated learning concept. So using blockchain to train your machine learning models on different systems without having to share the data. So for example, Melody Consortia is one of them, where there's 6 or 7 pharma companies that are participating, and they don't exchange any data on admin models, but the actual data, they're the stems of the model that get trained on that data, and the stems are combined on the central block, and that becomes your actual training, your actual main model. And that's how these companies are learning or exchanging their learnings between each other without having to exchange the data, which I think is a brilliant concept and should be utilized more.
It's like a pre-competitive type situation then, right? I mean, the way companies share data without, giving away their secret sauce.
Yeah, sharing their learnings, right?
Yes.
And helping, in different therapeutic areas, definitely. Yeah.
Great. Anybody else have comments about data?
I was just going to ask Ashwini if she has some thoughts on, as you mentioned already, the quality of the data. Do you see steps being taken to really try to make sure there's a good framework to have the right kind of data being put in, cleaning up the data, the curation part of it, and any advances that you're noticing or that you guys are working on?
Yeah, great question. So I would say not that we're working on right away, but I know a lot of companies, including bio, I think Arai, that is, that Rohit is part of, and there are many such companies that are using. I mean, that's just to name because he's here, but there are many such companies that are using NLP, or then using NLP is quite common to do this data curation. But a lot of effort has been put in within pharma companies and building this internal teams in order to clean up this data. But I still feel like synchronizing data is difficult internally, just because it's the same issue with getting data from publications. It's never same experimentation, never same concept, and so it's difficult to do that.
Great. So Rohit, you were able to join us?
Yeah, sorry about
to
that.
interrupt.
I was having some technical difficulties. Can you hear me now?
You're breaking up a bit.
Yeah, we can hear you.
Okay.
Okay.
So if the audio is not working properly, then I can try again. Otherwise,
I think it's good now.
Okay, cool. Okay, yeah. So I didn't get to the introduction, so I don't know where we are at. But if you would like, to talk about data engineering, if that be the topic still, then I can chime in on that. Okay,
sure.
Cool. Great. So as all of us know, I think the biggest effort in any AI or machine learning project is about the data wrangling or the data engineering, and that takes the most amount of effort and the most amount of time. If you ask any data scientist, they will say the same thing, that 80 to 90% of their time is spent, dealing with the data. And as we all know, data comes from a wide variety of sources, and especially, data, there is data explosion coming from a wide variety of sources in many different ways. So we need a robust infrastructure to think about data as an asset of the organization. And if we can make that mindset change, then we can begin to make investments from treating data as an asset perspective so that it becomes valuable in the business enterprise. So I'll stop with that and, see if anyone else on the panel has any comments on that.
Andrea?
So at GB Sciences, in addition to having our own data that we need to manage and integrate, what we've done is we've set up at a single interrogatable space in which we've put in traditional pharmacopoeias from 9 different regions in the world. And so there's an extra layer of complexity in that we're relying a lot on publicly available data. And as we all know, garbage in, garbage out, right? So every time we approach a specific pharmacopoeia that has been published, we are looking to filter, as well as we've been collaborating with experts in each of the geographical regions that we're working in so that they can translate not only certain words, but also we're layering, we take the base data that's publicly available and we're layering over it ethnobotanical considerations, including how do indigenous folks use specific plants and how do they use them in combinations, and layers that don't exist in the public databases get layered on top of that, so that when we have the full database that's available for interrogation, it's not just the compounds, the plant it came from, the geography. It's also layers of ethnobotanical information that were never put into the same space. And then we're taking, we're stacking it so that we now have not just one pharmacopoeia's worth of information, but 9 pharmacopoeias, and we're adding more with the idea that later when we want to talk about insights from convergence, you have to first make sure you've got a really solid database to start with before you're really going to get any useful information about the convergence. So that's kind of an extra layer of complexity.
Right. We look forward to hearing more about your plants too, on the next topic, Andrea. So anybody else have any comments who's in the data space? Ranga, Elmar?
Yeah, well, maybe I can add some flavor to this. So from our perspective, I think we are a hybrid model where we generate our own data for generating the biology and how that fits into the drug discovery paradigm. We do use the real world evidence and real world data, but one of the things that we have to deal with is the different kinds of disparate data that is required to connect the dots from discovery to development to clinical development. And so there are certain areas where you have 2 extreme segments of data, and we actually need to go out and either create or collaborate with somebody to generate that third set of data, which could be both a combination of structured and unstructured data that will then be brought in or work with a collaborator to get into a format that it is uniformly analyzable from one end to the other. It seems a little complicated, but it's relatively simpler once you have done this once.
Yeah, I guess so. It sounds pretty daunting to me. Any other comments about data? I know, Rohit, we were talking about historical data too when we had our pre-panel discussion, and how historical data just has its own set of challenges, and Andrea was sort of referring to that as well in her experience.
Absolutely. A couple of additional thoughts based on what Ranga said and what Andrea said. Ranga mentioned about unstructured data. So that's the other thing that how do you store all this data in a place which you can leverage later on? So how do you store it at the most efficiently at the lowest cost, so that when you are going to be running some compute or analytics on it, then you pay for that access to that data. So there are these new modern data warehouses which we have come across and which we have started implementing for some of our clients, which bring these new aspects to the table. And the other thing to remember about this data is that if you do treat it like an asset, then you have to think about custodians of the data, who's the custodian of the data, aspects about data governance, right? And then how to automate the ingestion of this data so that there is the least errors in the data. Like you said, garbage in, garbage out, right? So the data from a quality perspective, like it's not about the quantity of the data, which we have so much of, it's about the quality of the data. So how do we ensure the quality of the data in such a way that we get the best outcome when we do write the machine learning, algorithms and run the data sciences on top of it? And then the other thing, the last one, and then I'll pause, is also like all of us are concerned about cybersecurity. And, the new term which I heard this morning, data poisoning, right? So how do you make sure that your data is immune or protected from getting poisoned? And because if you can't protect against that, then it could lead to trouble later on. So these are some of the other thoughts around the aspects of the data, which is really like the key thing for any business enterprise. I think the most valuable thing actually for any business enterprise.
Right. Any other comments?
I have a fundamental question about the use of AI in the drug discovery process. So this is, in my understanding, a new process, new paradigm that many drug discovery companies are trying to embrace. So for the companies that do not have any foundation in AI-driven drug discovery efforts, where do they start? I understand that there is like no off-the-shelf AI, codes or modules you can buy and simply, start using. It has to be developed from in-house. Or is that not right? So what is, for example, if my company today wants to embrace the drug discovery and start AI-based drug discovery, where do they start? My question is to the entire panel.
So I could
start. Yeah. Go ahead, Rohit.
Yeah. So that's a great question. And I think that is where one other aspect which I wanted to bring to the table is that you do need some very good, first of all, it's about the data. So you must have some sources of data, whether it's coming from your LIMS system, it's coming from your labs, is it coming from external sources like somebody else mentioned on this panel? So first of all, what are the data sources? And then second of all, if you do have a combination of a data analyst with subject matter expertise, then I think that combination is very, very useful to have. A data analyst alone or a subject matter expert alone will find it very difficult to draw the meaning out of the data. And then, of course, the new skill, which is in short supply and going to be even in shorter supply, is a really, really good data scientist. So someone who understands how to draw meaning and insights from the data, and also to either use off-the-shelf machine learning algorithms, which there are plenty of, and I'm sure some of my colleagues here are already familiar with a lot of those, or you write from scratch. But fortunately, I think machine learning algorithms have come a long way, whether it is transformer learning or it is natural language processing. A lot of these are available, off the shelf, and you'll hear a wide variety of names, for these machine learning algorithms. So,
thanks.
Ranga?
Yeah, so this is an interesting question from our perspective and my perspective especially, because when we are talking about drug discovery, there are two sides to that aspect of drug discovery. And from our approach, at least at Berg, where we have one side of it is the target and the biology associated with it, and then the drug molecule that is being discovered, whether you use CAD or any in silico modeling for the target, and then how do you stick the biology to it. So there are two aspects to it. And at Berg, we use machine learning as well as AI on both sides of that coin, because we work with folks who are in the chemical space or in the, I shouldn't say the chemical space, in the drug development or drug strategy space, because it could be anything from small molecule, ASO, or any modalities, right? So we work with them, and there is plenty of AI and machine learning that is utilized. And what we do at Berg is we develop our own AI suite, which models the biology using ensemble modeling of sorts to then fit the biology to the chemistry. And so when we start iterating the chemistry, at each cycle, we are going back to the biology and seeing that the biology is reproducible to the endpoint that we need to then move it along the clinical development pathway, if that makes sense.
Thank you. Yes, that makes sense.
Any other answers to Elmar's question about where do you start?
I think Ranga actually hit it on the head, right? Because it really depends on what you need with it. So there are companies that believe they can identify new targets using machine learning tools and AI, and that's the approach they're taking. If that's what you're interested in, then yes, you need access to lots of data. You probably have some expertise in-house related to a specific target or some specific target biology that you're going to leverage. That's the subject matter expertise that Rohit talked about. And then it's a matter of accessing tools, and those you can find. Or you could be deploying it just because we're in small molecule space. If you want to design better molecules, you have a good understanding of the target, the second equation that Ranga was talking about. So I think it depends. There are companies that are doing the whole thing, and their shtick is that they're using AI to do drug discovery. But at the end of the day, they end up using the traditional tools that you have to do. You still have to make the molecules, you still have to make the ASOs, and you still have to do the biology to show it's panning out, because this is, at the end of the day, a prediction game. We're predicting something, and then you still have to make sure it works. So great question, and there's lots of different answers, but I think that's the two basic concepts of what you need to do with it.
Okay.
Great. Any other comments about the data aspect before we move on?
I have one more, Tina, to add. The last one is around, cloud computing or cloud engineering. So I think with the services that are available from the top players in this space, including what they are calling auto ML techniques now, I think cloud is definitely a consideration that anyone can make and get started very quickly. With there's no real need to kind of start building your own hardware infrastructure at your company anymore. I think it's a quick and easy start, whether you choose any of the top platforms, whether they be from Amazon, AWS, or GCP, or IBM, Microsoft. I mean, there's plenty of options available on that.
Great discussion. So why don't we move on to using AI and ML to understand disease? And actually, Ranga has a great story of how Berg approaches with the molecular profiling of human disease, comparing it to non-disease models. And so, Ranga, you want to start this discussion off?
Oh, absolutely. Thanks, Tina. So at Berg, we spent the last just over a decade of moving away from a traditional hypothesis-driven approach to knowing a target and a pathway to a very data-driven, human-derived approach. So basically what we did was invest heavily on generating human disease models, and then defragmenting the models to their molecular components of it. So we do high throughput, internally we do the proteomics, enrichment proteomics, lipidomics, metabolomics, and we outsource the genomics transcriptome because people outside do it better than we could ever bring it in. And so once we get that, what we specialize in during the building of models, when you look at a molecular matrix, it's always a slice in time. So what do you do with that data? You can't do much unless you enable it through a phenotypic characteristics. And so when we build the models, we actually, for each of our molecular framework, we have associated functionality built into it. And so we generate a lot of data, and we basically have a population that represents disease, and we have a corresponding non-disease, or one could say a control. And we developed an AI, and believe it or not, biological data, even when you generate genomic, proteomic, lipidomic data in a model system, it's relatively shallow compared to the large data pods outside. And so we developed a Bayesian statistics algorithm that is used to integrate all this data and then provide differentials between normal and disease. So we identify molecular drivers of disease, and then those become our targets, and then the surrounding linkage molecules actually become part of our biomarker development, so on. And then we work with the computational piece on the other side to go after drugging strategies. So we develop drug strategies based upon what is actually required, because it could be anything from a knockdown to a PPI to actually eliminating the RNA. So depending upon what that therapeutic endpoint looks like, we go out, get the experts and help us develop the drug at that point. So we have small molecules, we have large molecules, we have antisense oligonucleotides. So as therapeutic modalities that we are pursuing internally.
Right. So you mentioned that you start with a human, right? Human in vitro or the human model, and then you end up like back validating into an animal model that you can use, right?
Yes, actually, yes. It's a very cyclic process. So even within the in vitro setup, once the data is generated, we validate the phenotype, go back and make sure that we are seeing what we need in terms of a therapeutic efficacy supporting endpoints. And then one of the best aspects about going from a human disease and reverse translating it into animals is because the target itself is coming from the humans, we know that it is relevant to the disease. And so it makes it a lot simpler and easier to look through the spectrum of preclinical models. So it doesn't have to necessarily be a mouse or a rat. It could be any number of different animals which shows specificity in types of the phenotypes that are affected for the target. And so then we go after those kind of models, preclinical models, to go and validate the impact of that particular target in affecting that specific phenotype.
Right. So that's a really a translational approach to your animal models, right? You're not looking for the specific gene that's knocked out in animals and humans. You're looking for the phenotype that captures it.
Yes, absolutely. And you struck on a very interesting aspect, Tina. Ultimately, when we get into the later stages of clinical development, the genes do indeed help in moving towards a targeted aspect of it. So once the phenotype is, once we identify the phenotype, it's much easier to go and look at the genotype that is driving the phenotypes. So it's more of not hunting in the genes for a cause, but actually saying, okay, these are the causes, what are the gene pools that actually is dictating and driving this? And so it's from our perspective, it has worked really well.
Right. I find your company's approach really just fascinating because you deal with all kinds of test article classes, and you're sort of agnostic in your whole approach. So I think it's really interesting. Any other comments about the disease profiling or comments on what Ranga said?
I have a question for Ranga. I've always been a fan of Burge's approach, definitely. What are the challenges? I've been curious, what are the challenges that you came across in trying to build these kind of interactions, going from phenotype to genotype, so on and so forth, that you were able to overcome?
So it doesn't work linearly. So it requires that's the biggest issue. You think that, it's the P53, especially in oncology, everybody goes to the KRAS, P53 and the regular culprits. And what you find out is that it's not linear and it requires a lot more data generation at the back end. So we use things like CRISPR knockouts and other gene manipulations to then fine tune on the genetic basis to make sure that the area that we are focused on in developing these the models itself and then the validation part by the time we get into the clinical. Especially the preclinical efficacy validations are extremely important, whether we are looking at oncology or cardiovascular and metabolic disease. And so it's a lot of work, and sometimes the answers are not very straightforward, and so it requires us to go back to the drawing board and figure out where we are missing something very obvious. A couple of times we have had wrong hits in the sense that we have gone one way and realized that the answer is completely somewhere else, and it was just happenstance, serendipity, and we got us back to the right track. So I think those are normal trials and tribulations in any drug discovery and development programs. It's just that we tend to generate data in a high throughput manner to assess and address most of our questions, and then let the data drive our hypothesis for each of the steps.
Thank you. That is super helpful. So would it be fair to say that depending on which therapeutic area you have more data or training on, that you have better results, and the translation of that towards other diseases is not really linearly applicable? Would that be fair?
Well, you hit upon a very interesting aspect of it. So just to give you an example, we actually have a target in oncology that we found out has implications for a neurological indication. So we actually had to diverge and we were already developing a small molecule, and it was just a divergence of scaffolds to get an endpoint that was specific to oncology, whereas the other scaffold is going towards a neurology development. So one of the things that is extremely important is the cycling aspect. So every experiment, once the initial modeling is done, the progressive data collection in terms of validation, the data is cycled back to the original data sets so that, we are enriching the data as we go along so that subsequent machine learning and other computational activities, the granularity starts increasing on the outputs because of the added data sets. But the good news is these are all data that we are very diligent in going after the kind of data that we want to generate and the specificity of that data set.
Thank you. And the reason I asked this question is because I think a lot of challenges that lay in this area are because most of the companies focus on certain therapeutic areas. The data is rich in that, and so you have more accuracy around it, and the translation to other therapeutic areas like rare diseases could be a problem. And so how easy it is to do that kind of translation from one therapy, that was a curiosity.
Yeah, so we haven't gotten into the rare disease space as much because rare disease ultimately, most of them are genetically driven, whereas because we are building holistic models. So the Berg model for disease itself is very holistic. So we get multiple biospecimens from multiple organ systems, including the blood and everything else. So the network biology is more of a systems engineering component of it rather than one, if you're developing it's not like we are developing something for the kidney or the liver or the heart. It includes all of those organ systems up front in the model building.
Thank you. Sorry, Tina, I hijacked.
No, that's fine. Those are great questions and great discussion. So why don't we move on to, I want to make sure we have time for the designing and optimizing novel drug candidates. And actually, Andrea has a great story that's very unique, I think, in the phytochemical space. Kind of scary sounding in the beginning, but then she explains how she simplifies it for regulatory purposes. So Andrea, why don't you tell us about GB Sciences?
Sure. And so actually, I'm gonna give you. We have a sort of a 1.0 and a 2.0. So GB Science's goal has been to create these multi-component drug candidates where we can demonstrate synergy. And in our 1.0 version, we were really starting by looking at within the cannabis, different varieties of the cannabis plant, which includes hemp and all that stuff. And in the early days, we were using sort of off the shelf machine learning type tools to help us with things like predicting efficacy based on network pharmacology. And we were using tools for looking at the specific binding pockets in the receptors that were our targets, and using that to evaluate in a mixture how are these different how is the binding affinities likely to relate to the physiological outcome and things like that. Since then, we've now broadened our scope, and as I mentioned earlier, we're not just looking at sort of those compounds within the cannabis plant, but we've taken on all of traditional medicine, which does sound like madness, but someone's got to do it anyway. But the sort of the big idea behind that was that we thought to ourselves, if we can get all of these traditional pharmacopoeias into a single interrogatable space, and we can layer on information that other people aren't necessarily trading with, the idea would be that you figure over evolutionary time in different regions, people have been treating different symptoms with plants. So there's got to be some efficacy there, right? And so if you think about this on different places in the planet where these systems evolved, people were using the plants that were closest to them, and they're all sort of separate experiments. So the idea then is, if you want to try to predict the efficacy of novel formulations based on these kinds of compounds, if you look at the convergence, where do they overlap? There should be some truth there that helps to predict the efficacy of these different mixtures. And then to get at the sort of to really get to the next level of how do you decide what to put in mixtures, that's where we really had to dive into not just but the botanical considerations, but also the epistemology that underpins how traditional medicines are put together. The fact that there is a rhyme and reason about why different compounds are in the same formulation. And so it's having to create this new space that's got these layers, interrogate it, and also start to build up, through these machine learning algorithms and pattern recognition, the ability to predict combinations. And we're looking for minimum essential combinations from this vast data set. The other thing that is sort of unique about our approach is that we've done a very modular design to our platform, such that you can start by saying, I have this single plant. I'd like to figure out what it might be good for, and let's predict some possible minimum essential mixtures that we could test for certain diseases. You can also come in and say, I really care about this specific clinical indication, and I'm working in this particular region. Let's find things that are in this region that we could use. Or you can say let's interrogate the whole space. You can also use it to find botanical substitutes for other plants. So say you're looking for pain relief in a specific region, and a lot of the good examples are not in your region. You can also use this program to potentially suggest new traditional medicines for a region where they don't have one. Pain's probably not good on that area. But I guess the idea here is that there's so much data inherent in these analyses without machine learning and without trying to get to that sort of deep learning. There's no way that you could ever predict these potential combinatorial treatments and to evaluate their drug target indication efficacies. So that's kind of where we're coming at this, and how we use AI, we think, in fairly novel ways. Tina,
you're on mute.
So any other comments from folks about using AI for chemistry, for designing and optimizing your compounds? Stephanie?
Yeah, absolutely. I mean, as I was saying before, I come from a protein degrader company. Do you guys hear me okay? I know. Okay, great. So I'm going to speak in more broad terms because I'm a biologist and I'm not a chemist at all, as much as I love our chemists. But I think something to appreciate about where we are today, our technologies, is that when you are doing anything targeted, it comes down to the chemical environment of the protein and finding the ligand binding site. So there's technology such as AlphaFold, which outperforms any other machine learning technology that we had previously when it comes to predicting the structure of a protein. And it uses just the primary sequence of a protein instead of using like a de novo or homology approach to it. So as a biologist, as we all know, the drug discovery process, it's long, it's costly, and takes about, I think, 12 years to get a drug approved. So if we have these technologies that can help us characterize lead molecules and give us the most accurate approach to targeting a protein, it can not only expedite, but also save money and potentially reduce off-target effects and whatnot. So just something as a biologist I appreciate.
Right. Manish, you have anything to add to this about how you guys, how you use?
I think I think we probably use some of the same tools Ashwini is developing at Millipore Sigma as well. So there's a couple of aspects where we're using machine learning and AI to help with our clients' programs. So we do a lot of small molecule synthesis. So in the early stages, when it comes to designing, in silico design, the traditional methods still work and people still use them, but people are leveraging quite a bit more of machine learning techniques. We're doing the same thing. It's more deep learning techniques to kind of try and justify or better predict the kind of molecules that are going to be showing you the kind of activity you want. So that's one aspect of it. There's lots of tools out there. There's some tools that are commercially available. There's some tools that we write in-house to kind of do that. In terms of collecting data sets for this, though, it's still a lot of public data sets. You're looking for homology models. You're looking for structural information that's available. You're looking for SAR data and synthesizing all of that and using the algorithm to kind of help generate some predictions for you. The other aspect where we are using it quite a bit more is to really help predict some physical chemical properties. So people working in the CNS space, they're very, very keen to know, well, is this going to get past the blood-brain barrier? So we can start using these tools, looking at the data we have generated and public data sets on molecules to start predicting the behavior of the molecules we're designing and synthesizing for them. And looking at other, what's going to be the half-life? What's going to be the residence time on target if you have some data on the structure of the target that you're going after. So we do that as well. And then the third place where we're using it quite a bit is completely opposed to, it's not the drug discovery part, it's just the execution part. So synthesizing molecules, we have target molecules, retrosynthetic analysis. So basically taking away the effort of one or two chemists looking at a structure and just leveraging the information that's been collected over years to kind of define what's the best synthetic route to get to this molecule, predicting yields, understanding what's going to be safe. So these are all things that we're now finding are becoming really enabled with some of the tools that are available. And it's not something that we have to build in-house. There are companies that are doing this routinely to provide these techniques and tools for us to use now. So it's making a massive difference in speed and how quickly we can get to some of these answers and how we can execute and make the molecules. And then eventually you get them back in the biologist's hands, like Stephanie, to actually get the testing done and understand if it's panning out. So.
Yep. I would like to add to Manish's point. Like Manish has put together, really comprehensively summarized, all the components. And most of the times, process optimization, retrosynthesis gets, it's really hard to tackle. Right, the data is so small, relatively, and plus there's no negative data to accompany what has failed. Everybody publishes only what's worked, which is a problem with reality. About, we can have on the negative data sets, but not to divulge here from the aspect that that's a critical element, right? And designing how to synthesize compounds and do so on and so forth with these ideas. At the same time, I think we talk a lot about AI ML, but the computations that have served us well throughout are equally integrated. The descriptors that are coming from there are the ones that are driving the AI ML processes. So I think the companies that have succeeded, the technologies that have succeeded well, are actually a blend of both. you really have to know where you could use AI/ML. Do you really have the data that's required for that? Can you really use GAN approaches to address that particular problem? You can't really, and to be honest, in the scientific world, we don't have enough data to train all these models. So I think aspects where you can do generative design very early on, you can use some of them for admin prediction where you have some data, but there are some elements where you have to rely on compute. And actually a good blend of those is, I think, is quite accurate, I would say, in the success of these.
I couldn't agree more, Ashwini, and that's why it's our computational group that does this. There's nobody else within our organization that can do this. They have the knowledge and they have the existing tools and what they can leverage there. So, well said.
Great. I keep losing. Sorry, I keep losing my network. So excuse me if I'm garbled, but go ahead.
Oh, sorry. Okay. So I actually have a question building off Manish and Ashwini's points. Just a question to the panelists here. Have you guys used any AI models or combination studies to address like tox studies or for animal model predictions? And just out of curiosity.
There's an existing suite of tools that people use routinely to try and predict genotox, for example. So there's the ICH-M7 guidelines, there's Derek, LASA, Nexus. These tools have been available. I don't know how much they're leveraging machine learning as much as it's more computational and based on existing data sets. So they are still making predictions. At least for us, we haven't been using AI tools to kind of help enhance that or speed that up. But I'm sure there are people working on it. And maybe somebody else on the panel knows somebody who is doing that.
Yeah. I mean,
I'm not aware of it, but someone else, go ahead. Sorry.
Yeah, I was going to say, not specifically any tools per se, but I think most of it is driven from the data for AdMeTalk specifically. And so these consortia and bringing that data, the knowledge together to build upon, I think that would be, that is the approach that I think worked the best at this time.
Thank you, guys. I appreciate it.
You probably have seen more than anybody else when it comes to talks, if there is something out there.
Yeah. Well, we there's certainly a lot of efforts to try to improve predictive tox, right? In vitro prediction, and we're not there yet. And at the rate that FDA moves, at the speed that FDA moves, I don't think that's going to happen in my lifetime. So anyway, maybe I'm just a skeptic. So why don't we start talking about some future trends in these areas and other breakthrough technologies that you guys think are going to move the needle, so to speak, in the drug discovery space.
So maybe I could chime in with the first thought on that. And I've been kind of, learning more and reading up on it because it's still in the nascent stages as quantum computing. And I think currently we are using a lot of supercomputers and GPU, power. But I think to make it really fast, really efficient, and to be able to look at a really large amount of, combinations. There are some companies who are at early stages of experimenting with quantum computing. And I have a feeling in the next few years, maybe 3 to even 8, I think quantum computing will start coming into play in many different ways. So that's a little from the hardware side of things or infrastructure side of things.
And maybe I can add to the data acquisition part of it. Given that in the drug, especially in the clinical development space, we have always been dealing with the traditional data acquisitions, from the clinics. But the advent of technology, especially peripheral wearables and IoTs, and now things are getting so sophisticated. For example, a good example I can give you is there is an active effort in the Parkinson's disease drug development space to look at movement, and Sage Bionetworks is actually, there's a consortium going on looking at wearables where people's actual tremors are being captured in real time over a longitudinal segment for several days. And they are using AI/ML to then fine-tune when the drugs are on and off in terms of prediction. And that is now getting into the mainstream. And things like Apple Watch, where you are getting your ECG, the cardiogram. Now, how good they might be from a clinical development space, I don't know yet. But I think the ability to capture more data in a non-invasive manner using all kinds of electronic and chemical sensors is getting mainstream. In fact, only a couple of days ago, I read about a study where they use green light that penetrates into the wrist using an Apple Watch-like thing to actually activate a protein in a preclinical model. So those kind of technologies are actually coming up so fast that in the next 5 to 10 years, data acquisition as we know it is going to be completely different, and all sorts of data sources is going to be useful in the drug discovery and development space.
Yeah, it's kind of scary, isn't it, Ranga, when you think about people being able to hack into your all of your health aspects. Anyway. Yeah,
absolutely, and that's where what Rohit said about cybersecurity and data security is so important. I think we are going to get there because personalized data and there is massive movements. I know in Europe where there is individual ownership of their medical data. So how do you balance off the security of medical and health data and the ownership of data to an individual who may not be savvy to electronic equipment, and how do you gatekeep that? So absolutely, we are getting into uncharted territory, and there's going to be a lot of discussion.
Yeah,
I have a somewhat scary prediction for the panel as well. So historically speaking, big pharma has always considered the appropriate way to look for I think that this AI machine learning is going to transform the way we think about drug discovery, because big pharma has sort of collapsed drug discovery into an exercise where you're trying to have a single ligand for a single receptor or a single drug, single target. But there was actually a really cool paper that came out that's been talked about a lot a few months ago that talks about the fact that even though most drugs gain their approval based on the idea that they have a single target, almost all drugs that are out there have polypharmaceutical effects. And so there was a practical reason why we had to be reductionist, right? There, back in the pre-computer days, pre-AI days, in order to have anything be meaningful above noise, you had to sort of have a simplified test case so that you could understand the results. But now that we have these new technologies available to us, perhaps more and more companies are going to start to think about all of their drug discovery activities as being polypharmaceutical in nature. So, whether it be people like us or just, general big pharma folks may sign on at some point. So that's kind of one of my predictions.
Yeah. And I think a lot of that we even saw with multi-drug, combination drugs, and that have been coming and been used in that realm. But what I find most exciting, and I've already shared about federated learning, which I'm super big fan of. But apart from that, what attracts me the most, I think, on the clinical side is the digital twin. The idea that you'll be able to run an entire clinical trial in silico, it is fascinating to me. I would like to see that happen quickly. So that's where I think the future is.
I have a more fundamental view of the future of the drug discovery, especially with AI in it. As a biologist, it's fascinating how much multi-dimensional data we can handle. These days we're looking at proteomics, genomics, RNA-seq, CRISPR, single-cell proteomics, it's vast. The landscape is expanding. So, but when you look at the AI perspective, it's as if it's a boxed way of certain things. You're kind of zooming into a section of this drug discovery. You're oversimplifying things and trying to put some data and then predict something. So I would like to see a future of drug discovery where the AI component can not overly simplify, but accelerate the wealth of data into driving decisions, into driving these predictions. You know, the AI, I mean, in an overly simplified way, is data in, data out, right? So the data component is very essential. So the ability of the AI as a mechanism to absorb more data points, the data acquisition that Ranga mentioned is very important. And of course, that's a heavily machine and technically intensive exercise to enable AI to absorb all of that data. So it's a wish that, you know, AI can catch up with the speed of the biological development here, and also it's a reality call. Is that really possible? And I think the biology and the AI, I believe, like the two parallel highways, and they kind of have to go at the same speed so they can benefit all of each other. But I don't know if that is, which one is faster. I mean, is that really possible that AI can take into account all of these various disparate data points, proteomics, genomics, CRISPR, you name it? So it's just an open call or it's a statement.
So, Elmar, I just want to add something here, and this is in context with where we are right now. We are on a virtual screen thanks to COVID and the 18 months of this thing, of being sequestered in our respective rooms. But one of the things that diametrically changed the way clinical trials are run is, even within a span of 18 months, we have had to adjust our clinical trial. You know, it was ongoing, and all of a sudden, the need to pivot to significant pivot. I'm not talking about, we are going to shut down certain data capture points in a clinic or something like that. So literally working with a CRO to go to the patient rather than the patient coming into a hospital. So this is like a huge, structural component of it, where the data source now comes, when you think about it in a real world setting, coming into a hospital, going through, taking the history, data getting entered into a ECRF, and now you're completely remote, people going into the patient's house, and the data acquisition is occurring via various different sources, right? So telehealth and everything else in the COVID-19 situation has changed and disrupted that it's going to get more and more unique, where the traditional aspects that we know it in terms of clinical design, and I completely agree, with Ashwini's point of in silico, for the level of density, high density molecular data that we capture from our clinical patients, pretty soon, if we have sufficient numbers, there is no reason why we can't use those in a dynamic fashion with the right kind of machine learning tools to model systems that way. So I think we are going towards that reality, but I just think that it's going to take us a while, and more so from Tina's initial note saying that, we all these activities eventually has to hit the regulatory aspects of it. So we may be going at warp speed, but then there is this hard break that we hit because the regulatory agency is going to get, so what? Show me the proof, and whatever they are convenient with or comfortable with is what is going to ultimately help in navigating through that particular piece of the approval process. So it's there are so many different challenges. I think there are aspirational aspects to what we are discussing, but then we eventually are hit with the reality of the daily lives that we lead and the limitations that will be posed. So I'm sure we are going to make headways over the next decade, but I think from a drug development, especially in the regulatory space, once we reach a certain level or extent of development, it still will require certain traditional paths that may interface with the technologies that we are talking about. But I don't see that to make radical changes within the next decade or so.
Yeah.
Right. Yeah. Maybe my comments about FDA were a little unfair, but in the rare disease space, they do make exceptions, and so I think the rare disease space is really helping us pave the way to sort of non-traditional approaches to drug development and drug approval.
Oh no, Tina, my statement wasn't meant to be this thing about the agency. For example, they have been extremely good to Berg, because when we started about in 2012 for molecular captures of the pharmacodynamics of a drug in a phase 1 clinical trial, that was significant impact on the patient itself in terms of in-hospital stay. And so, but they signed off on it, and at the back end of it, we actually gave them a clinical study report that has all sorts of things that is non-traditional. And progressively, when we went into our subsequent INDs for phase 2 and beyond, the progress with the agency and the discussions has been much easier given that historical perspective. So as long as we, from a development perspective, are able to generate the packages that support everything that we are doing, they are extremely open and and guide us in the next stages of clinical development.
Right. I guess my comments were more about how long it takes to get a guidance approved,
exactly.
Right?
Yep.
And years.
Yep.
Anyway.
But then again, there's been another, on the regulatory side, there's been another trigger too, which is COVID. So certainly, as we've all mentioned, the very way that trials have been monitored has changed significantly because of having to isolate and all this data that's coming from, on the patient's home and through digital means. I suspect that means that FDA will be responsive to that because they know that that's a real trigger in our world too.
Can I play devil's advocate here just a little bit? So much of the excitement in the AI space is on the clinical side. When you have a drug, a supposed experimental drug, and then you're designing your clinical trial, selecting target population that would benefit the best out of the drug based on some key markers, right? But let's not forget that the drug that you are testing in clinical scene comes from the drug discovery that's done way deeper in the labs that takes years. So we want to accelerate that too. And, if you look at the entire span of the drug discovery, there is the early drug discovery, preclinical, and then the phases. So in my understanding, certain parts of this drug discovery process benefit from AI-driven or progressive technologies more than the rest. And the phase, the part of the drug discovery that benefits the least is the early drug discovery, where there's a lot of biology going on, a lot of chemistry, docking, compound after compound. That part, in my understanding, benefits the least. The clinical side, the patient, the person, the toxicology side benefits the most because for whatever reason that is, maybe I'm wrong, but maybe that's a low-hanging fruit for the AI field. So do you think I'm right? Do you agree with me? Or otherwise, do you think one day this will normalize and spread evenly to the entire span of drug discovery?
Ranga, I know you want to comment, but I would like to comment.
Ashwini, why don't you go first and then I will take this.
So, great question, Elmar, but I sort of see it a little differently from the perspective that clinical trial and all, we have a lot of data that we can leverage to do some kind, to analyze it and build your predictive models, right? Getting that data from the regulation, all of that's different realm. But once you have access to it, you have enough amount of data to train good models. The problem with discovery is that it is a really difficult problem to solve. To teach chemistry to a machine, to teach biology to a machine, it takes years for us to become, I don't even want to say expert in this field, but to gain enough knowledge to do right predictions. So I think it is this fundamental difference in the problem that we're solving that is quite different. And also the data that you need in order to solve that problem is quite small when it comes to discovery. So once we have enough data, we will be able to solve it. But I think at this time, it's like looking for data in finance, and they use AI ML quite often because they have enough of that to do so. So I think that's the difference from that realm. But to address your previous point there on can these combine, which one has to be faster, biology or AI? I think this is the beauty of AI ML, that it's actually brought it all together in the same realm. It's bringing chemistry, your phenotypic data, genetic data, all of that data together when you do small molecule prediction or large molecule prediction, so that your success in discovery is much higher and you fail less in the development. So I think that's the beauty of all of it. So I'm going to pause with that. And
So I'm just going to be very brief here. Elmar, I just have a different outlook to it, because we fundamentally use because discovery biology is much shallower than all the data, other data parts in the clinical. We actually developed a Bayesian math-based algorithm to actually do the discovery biology in the preclinical space. And here is what we have been able to achieve it took us about 10 years. I'm not going to this thing, but it took us just over 10 years. But we have been able to connect the dots between discovery biology and a phase 1, and now we are honing in on a phase 2 analysis where the pharmacodynamics of the molecule is being linked to discovery biology. And the answer is it possible? And I'm saying from our experience, yes, it is. Here is the thing. Does one size fit all? No. The maths are different, but it is possible to link those two. And what we have moving towards is reducing the risk in the translational valley of death, where the preclinical data supporting the early stages of clinical success by collecting data that is extremely focused. So we are not randomly going and saying we are going to collect data, but focused on what we have in the discovery and what do we need within the early stages of clinical trials so that we can link that data. And so we have the toolkit, we have developed the toolkit, and I'm happy to say that I have independent validation of the discovery biology in a phase 1 and now in a phase 2, not only on the biology of the small molecule, but we have used traditional analytical components that is readily available to also pass through the data for understanding AI, the adverse events and other clinically relevant endpoints that is necessary for the clinical development. So it's getting there. It's not easy. It took us a while, but it's doable.
Thank you, Ranga.
So I had just a quick comment. I agree with both of the. So I'd say, Elmar, way to play devil's advocate.
So I'm gonna.
Oh, we have to cut off. Oh, sorry. All right. So just quickly, I was going to say there's another thing that's not that I also agree that over time. AI and machine learning will be applicable across all phases from discovery through clinical. But one of the other things that isn't as scalable is it's not just that in discovery you have shallower data sets, you also have smaller companies with smaller budgets. So perhaps the preponderance of data, preponderance of the use of AI in clinical is also being favored in larger companies that can afford it, right? So that's just another comment.
Okay. I told you guys we had a passionate and enthusiastic group of scientists, and I was right? So I'm going to have to stop. I'm going to have to stop us there. And thank you guys so much for an exciting panel, and I wish we could do it again. So I think Sam actually has a few parting words, and also we have a raffle later. So Sam?
So again, thank you guys so much for participating in our interactive panel discussion. I want to thank you, Tina, for organizing this. The brainstorming sessions was very insightful. This panel was recorded. Very soon we'll edit the details and we'll publish it on our YouTube page so we can all watch it again. I want to thank Dr. Andrea for her time and commitment throughout the day. She's been up since about 4:30, 5 a.m. Pacific time, and we appreciate her efforts today and her remarks. It doesn't go unnoticed. And there is just a quick raffle draw I want to do on behalf of our team to reward our attendees throughout the day. So here are some of the tickets that we got. And I'll start with the $100 Amazon.
Sorry with that. I had a technical difficulty with my cell phone. So
I'm just going to draw a couple names here. First,
wonderful. Wow.
Yeah, so we got the attendees that visited the booths and collected the passcodes. We got their names from the emails.
Definitely. So, it seems like the sponsors definitely were kind of guaranteed a slot.
Yeah. So for the first $100 Amazon gift card, we'll go to, and sorry if I mispronounce your name, Hideyoshi Fuji from Estella Pharmaceuticals. So Mr. Fuji, if you're around, we'll also reach out to you for your $100 Amazon gift card, which will be delivered virtually, of course. The second prize for $200 Uber Eats gift card, just here, roll it up. This is Teresa Tribble from Ingen XYZ. So Miss Teresa Tribble will reach out to you for your $200 Uber Eats gift card, which should last you about a month's lunch and dinner. And then finally, for our 5 Series Apple Watch, and the winner of the Apple Watch from the PDDP Summit is Dr. Chin Ni Chin from Merck. Dr. Chin, congratulations. We will reach out to you to send you this gift. Again, thank you all for participating at this PDDP Summit. We do also have day 2 of our summit happening tomorrow, starting at around 9 a.m. And for that, chairperson will be Dr. Finith from Roivant Sciences. So again, thank you, Dr. Andrea. Thank you, Dr. Tina, and thank you, everyone, for joining us today.
Thanks, everyone. Bye.
Thank you. Bye.